All right, revising for your GCSE biology exams? Then you know just how important these required practicals are. Today, we're going to completely nail required practical 10, investigating the rate of decay. Let's make sure you are 100% ready to ace any exam questions they throw at you.
So, here's our game plan. First, we'll get a solid handle on what decomposition actually is. Then, we're going to walk through the milk practical step by step. After that, we'll look at how to analyze your results and draw a proper conclusion. We'll also cover the science behind it all. And finally, we'll shine a spotlight on the exact skills that examiners are looking for.
Okay, first up, what on earth is decomposition? And to get our heads around it, let's start with a question you've probably wondered about before. Why does milk go sour?
Turns out this everyday problem is the perfect way into our topic. That whole process of milk going off, well, that is decomposition in action. It's biology happening right there in your kitchen.
So, for your exam, you need the official definition. Decomposition is the breakdown of biological material by microorganisms. A great way to think about it, though, is like nature's own recycling system. It breaks down all the old stuff and recycles the raw materials back into the ecosystem.
Now, this process doesn't always happen at the same speed. The rate of decay can change a lot depending on the conditions. The three big factors you need to know are temperature, water, and oxygen. And for this required practical, we are zooming in on just one of them, temperature.
Right then, let's dive into the practical itself. The whole point of this experiment is to investigate how temperature affects the rate that milk decays.
Okay, before any experiment, we need to be crystal clear on our variables. The independent variable, that's the one we change on purpose, is the temperature. The dependent variable, the one we measure to get our results, is the time it takes for the pH to change. And, super important for getting marks, are our control variables. Things like the volume of milk and the concentration of the enzyme have to stay exactly the same. Why? Because if they don't, it's not a fair test and our results won't be valid.
So here's the play-by-play. First, you set up your water baths at different temperatures. Next, you prep your test tubes with some milk and a pH indicator. Step 3, you pop a test tube in each water bath and let it sit for a bit, to get up to temperature. Then, you add your lipase solution, that's the enzyme that does the decaying, and you hit that stopwatch immediately. And finally, you'll watch like a hawk and record the time it takes for the indicator to change color. That color change is our finish line.
Great. The experiment is done and you have a bunch of times written down. But that's just raw data. The real science, the bit that gets you the marks, is turning those numbers into a proper conclusion.
Now, listen up, because this is a classic mistake students make. We measured the time, but the experiment is about the rate. They're not the same thing. To get the rate, you have to use this formula. Rate equals 1 divided by time. It makes sense, right? If a reaction is super fast, the time is really short, and 1 divided by a tiny number gives you a big number, a high rate.
And here's what a results table would look like. You've got your temperatures, the times you measured, and that crucial third column for the rate you calculated. You can already see the trend. At 20 degrees, it took ages, 900 seconds. But at 40 degrees, it was much quicker, only 240 seconds. A shorter time means a higher rate, and the math backs it up.
This is why we draw graphs. That table is okay, but when you plot the rate against the temperature in a chart like this, the pattern just jumps out at you. It's so obvious that as the temperature goes up, the rate of reaction goes up too. Being able to turn data into a graph is a huge skill for your exam.
So we've seen what happens. Now we need to understand why it happens. What's the science that explains our results?
The secret ingredient here is enzymes. In this case, an enzyme called lipase is breaking down fats in the milk into fatty acids. More acids make the pH drop, and that's what makes our indicator change color. Now, enzymes work best at a specific optimum temperature. As things warm up, molecules move faster, collide more often, and the reaction speeds up. But if you get too hot, the enzyme denatures, it loses its shape, and it just stops working. The rate of decay pretty much falls off a cliff.
And this isn't just something that happens in a test tube. Think about a compost heap in a garden. Gardeners are basically trying to be decay managers. They're creating the perfect warm, moist, and airy conditions so those microorganisms and their enzymes work as fast as possible to break down all that garden waste.
Okay, last section. Let's tie everything we've talked about back to the exam. What specific skills have we actually been practicing here?
I mean, just look at this list. By getting this one practical down, you are ticking off a whole load of different skills. You've got your hands-on practical skills, your math skills with calculating rates and drawing graphs, and those really important working scientifically skills like controlling variables and spotting trends in your data. This is why these practicals are worth so many marks.
So, I'll leave you with this final thought for the exam. To get those top grades, you don't just say what you did, you have to evaluate it. Think about it. How could you make this experiment better? Deciding the exact moment the color changes is pretty tricky, right? That's a source of error. To make your results more reliable, you should always do repeats and calculate a mean. Asking these kinds of critical questions is what separates a good student from a great one. Keep that in mind and you'll be in fantastic shape. Good luck with your revision.